assembly for HMP data sets, and that is impossible. So you just wish to, you will get more contigs, more longer contigs. So you don't even bother to try to get like complete genomes, a few of them. So you just try to get as many long contigs as possible within reasonable time using reasonable computational resources. And gene prediction, so if you have very long contigs, it doesn't matter, you can use whatever you want. But as I said, probably like 50% of reads cannot be assembled. It's quite, it's very common that 50% of the reads in a metagenomic data set cannot be assembled. So you will have so many short reads. You still want to do something for those short reads. So one problem is that can you predict, for example, protein fragments from those short reads? So it's still a problem, but it's really something new, I think. It's a new problem because you have never.
The reads cannot be assembled. Can they also be aligned to some already known species? Or you do that first before you do the assembly?
So there are two groups. One group try to map all those reads to the reference genomes. For those ones you can do, but you will still have a lot of genomes that you don't know. So yeah, you can still do that, but most of the cases you will still have a lot of short reads that you don't know. You cannot map them to the known species. You cannot assemble them into contigs. So you still have a lot of those short reads. But actually many, I really use a lot, too much time. But I think many groups they just try to predict the functions. Just use the short reads. They don't even bother to do assembly. So if you read papers, there are quite some papers just based on annotation using short reads. No contigs, no assembly. So, but definitely that's one of the problems when you try to annotate those short reads. You just try to predict the proteins or protein fragments from those short reads. And it's difficult, as I said, the signal is not really strong. So if you have long contig or you have whole genomes, probably it's easier to do the gene prediction. But if short reads, probably it's hard. And also they have sequence errors. So if there are sequence errors, they will mess up with the frames. So then you cannot really do gene prediction. Function prediction is really largely limited to those similarity search-based methods so far. And BLAST is the method that's often used because fast enough, but it's still not enough for the HMP. So HMP, I think all the similarity search is done in WashU, and they have to use some software that's faster than BLAST, that 1,000 times faster than BLAST from a company for that job. Because they claim they have a supercomputer cluster with like thousands of CPUs, and if they run BLAST, and to BLAST all those reads against NR database will take for 40 years. So that's the reason they don't go with that BLAST search. They get I think it's called MBLAST or whatever. So that's really a software from the company. So it's about like they call it. They claim it's about 1,000 times faster than BLAST. But I don't really know because it's commercial software. Yeah, but I really want to say what's the performance. I mean, if you definitely can come up with something that's like 1,000 times fast or even faster than that, but you need to when you talk about the specificity or sensitivity, you don't know. Usually faster ones will miss a lot of those not so similar ones. And another is the binning of metagenomic sequences. The meaning of binning is that even though you may not be able to assemble individual genomes from a mixture of DNAs, but if you can cluster those reads from the same species in one cluster, so that you can do something, or you can do something from there. So if you can do that. So this is just some interesting problem in metagenomics. And there are some interesting methodology developments especially a few years ago, there are quite a few new methods coming out just for that purpose, to bin the metagenomic sequences, hopefully into species. But I think, again, the practical values is not really clear. So if you read papers, especially those large metagenomics papers, you don't really say that this binning is used, or in any sense. But if you already get a context, you can do binning. So big context, you can try to classify context from one species into a bin, so that you know they're from the same species. Because if you have long context, very long context, they have enough features for you to classify them into species. And of course, we need to compare whatever we learn, like protein families of pathways from different environments. We want to compare if like this pathway or this function is enriched in this environment. But there's still a lot of problems because again, the data is incomplete and the reads are short, so that the annotation are not perfect. You will get a lot of artifacts here if you are not very careful about the analysis you are doing. So, but definitely it will be very interesting if you can model or you can predict the interaction between different microbial organisms within one environment, or how those bacteria interact with the environment. Or if it's animal related, it will be how those bacteria interact with the animal host, like how the human gut microbial organism interact with human. So those will be very interesting topics to explore once we clean up how to analyze those short reads. So Megan, I just want to mention a couple of very interesting ones. I will not take long, I think. Megan is a software which was first proposed to classify reads, here not only 16S, but all the reads into different taxonomic groups, using short reads, all the short reads. So I will, it uses the lowest common ancestor algorithm, so if one read is similar to multiple groups, so you will take the lowest common ancestor of that several groups and use that one as the assignment, taxonomic assignment of the read. And but I think the authors, they expand the functionality of that Megan software. And they have like Megan 2, 3, and 4. So you can do like functional analysis or comparison between multiple metagenomes. And also, I think the good thing about this is that they provide visualization, very nice visualization of the differences and the commonalities. So this is the lowest common ancestor algorithm used in Megan. So essentially, if you have a read that's similar to these three, to some sequence from these three species, and probably all the similarities are significant, so you don't really know if it belongs here or that one or the other one. So you will just use the common ancestor of these three and use that rank as the taxonomic assignment of the read. And they can also do like a function do the comparison at the function level, like including families or pathways. I think this one they use the pathways or what? Oh no, this one is just for a comparison of multiple metagenomes. And also the right side will be all those assignments. And for example, if this one, I don't know what's the color of this one, but if you found like reads can be assigned to this species from one sample, then you will color that one, corresponding one. So this one, then you can look at this, how similar these two environments are based on the taxonomy. Or you can really look at the other aspects, like the functions, pathways. So I think the Megan 4, they incorporate this gene ontology into this tree. So that's Megan, and I will. Can I still use like 10 minutes, or you want me to stop?
Either way.
So
are you okay?
I'm okay. So I will just this one. So what we can learn from those metagenomic data sets. So this is just one new paper from Science, Nature Science. So this is just one application. So what you can do with metagenomic data sets. So the authors they look for those enzymes involved in plant polysaccharide utilization. So especially they're very interested in the cellulase. They will be involved in the degradation of cellulose. So, but you can see there are a lot of data here. They get like about 268 gigabase from cow rumen, the bacterial community in cow rumen, because the cow can digest those plants. And they found these many enzymes that have like carbohydrate activity. So they are involved in somehow in those process. So, but it does not mean all of them will have like commercial values, but at least they provide some starting point. Yeah. So, and also I guess this is a non-redundant set. But anyway, I think when you do metagenomics, you have a lot of data DNA sequences, but sometimes you can get something like this one, 28,000 genes, something you're interested, but from 268 gigabase of DNAs. So probably they just keep sequencing that sample. And you can look for like antibiotic resistant genes from metagenomic sequences. And you can look at the adaptation of those microbial organisms when the environment changes. And you can study, for example, human impacts on the ocean microbial communities. And these are 3 resources. MGRAST. I think it's the most commonly used one, and they have analyzed this many data sets. So if you're interested in a management data set, you can go there, and they have all the original data sets, all the analysis results. Camera was actually initiated earlier than MGRAST, but I think it's not as active as MGRAST. Because MGRAST, Volker Meyer is involved in the MGRAST, and he's very active on that project. IMGM, it's really kind of small as compared to the other 2. They only have several hundred metagenomes. And it's really based on the IMG, the integrated microbial genomes. So they just extend that one into IMG-M. To the metagenomics. And I don't really think I will go through the methods we have developed, but we're interested in coming up with new algorithms, new software, so that hopefully we can make more sense of those metagenomic sequences. So fraction scan, the one we developed for the gene prediction from short reads, error-prone reads. And this is a very new work we have done. We try to identify those CRISPR elements. So probably many of you don't know really what CRISPR means. It's just you can just consider that's a bacteria's immunity system. So it has nothing to do with antibody, antigen, but it's really some mechanism that bacteria have against the virus. Bacteria, they also need to fight against the virus. So those will be the CRISPR elements. So they have genes, Cas genes, involved in this pathway, but you can just consider those CRISPR elements. They're just repeats and spacer units. So those repeats will be the DNA regions in the bacteria, and those the spacer sequences they're actually taken from virus. So when the virus invades the bacteria will take in those pieces from the virus genome and insert those virus fragments into their genome. So later on, RNA can be produced from those repeat spacer units, and those transcripts can guide the fight against the virus. So it's a very interesting, very interesting bacterial immunity system. So, but the problem is that it's very hard to assemble those ones because you have so many repeats. So if you just you just run super de novo, you cannot really annotate those repeats very well, or those elements. So that's we come up with targeted assembly approach for those elements. So we will try to fish out all those rates that contain some of those repeats, and then we just do assembly only on those pulled rates. So that's the reason we call it targeted assembly. So the assembly targets at the CRISPR. So using the repeats as tags to fish out the rates. So by doing this, we can really assemble a lot better those CRISPR repeat spacer elements. So once we have the assemblies, we can use the spacers to do other types of analysis. We can look at the dynamics of those spacers among different human individuals, and one of the results shows that they're very dynamic. So, for example, in one of your bacteria, it has a set of spacers, but if you have your sample sequenced again in a couple of months, probably most of the spacers will be different. And that indicates new interaction between, for example, between the bacteria and the virus. So we can use those spacers to trace the interaction between the virus and also the bacteria. And also that helps us to hopefully to investigate on for example, the viral exposure of human beings. But it's just something very interesting, and we have this methodology published in that it's accepted in PLOS Genetics, but we're really looking forward to analyzing those spacers and try to figure out, do something about the virus and the bacteria interaction in human bodies. So something faster, but it's only like 150 times faster than BLAST. Not close to 1,000. But at least we can guarantee our method does not lose that much sensitivity. So we do get something from metagenomics, but there are a lot of artifacts. I did not mention too much of these ones, but like the bias of 16S RNA surveys. So if you're not aware of those bias, you may make misleading, at least misleading. You may have misleading conclusions. Or if you are not aware of the annotations, many of the annotations are not perfect, and you draw some conclusions based on those annotations, then you will also make mistakes. So we did have a couple of publications on those artifacts. So if you're interested, you can try to find our papers. But this is just a little review paper we have that's the title of the review paper we have. So if you're interested in the challenges in analyzing metagenomic sequences, and if you're curious about the artifacts, you please read that review article. And I'm free now to take any questions.
Question? I have another question actually. So can you briefly overview the future clinical applications of this metagenomics area?
So people call we need to do personalized That does not only include the human genomes. Consider the variations among different human genomes, but also the variations among the other genome, the microbiome genome. But I think it's still too far because of the huge variation you find in the microbial communities among in the population, and also like my community probably will be different, slightly different tomorrow as compared to today. So it's very hard to get the information if you have such large variation. But I think there's some work out there indicating like some bacteria, they are involved in this particular disease, or some bacteria involved in the obesity, some bacteria involved in the inflammation, whatever, different types of disease. So I think they will be useful, but I don't know. I don't know. Not yet.
All right, let's thank our speaker.
Thank you.